Imagine engineering your own bones to mass-produce a biological defense force, a factory right inside your marrow, pumping out millions of cancer-hunting cells every single day without needing constant treatments. Your body already has billions of immune cells constantly patrolling your bloodstream, searching for damaged or abnormal tissue. Yet tumors still manage to grow right past them. Cancer survives because it adapts. Some tumors physically hide from detection, while others release chemical signals that literally put attacking white blood cells to sleep, shutting down the body's natural defense mechanisms. Overcoming these cloaking abilities requires a targeted genetic upgrade to the immune system that goes beyond the limits of our natural defenses. That realization led to CAR T therapy. Doctors extract a patient's T cells and install genetic targeting software, training them to lock on to specific cancer markers. This process requires pulling cells from a patient, transporting them to a lab to be engineered, and infusing them back. Every batch is custom-made from scratch, taking weeks and costing hundreds of thousands of dollars, creating a massive medical bottleneck. But the biggest hurdle is biological. While engineered T cells excel at clearing blood cancers, they hit a physical wall when they try to penetrate solid masses, like breast, lung, or pancreatic tumors. T cells simply aren't equipped to break into that kind of dense, hostile environment. To fight solid tumors, researchers had to find a different biological strategy. Enter the macrophage. Unlike T cells, these massive white blood cells naturally burrow deep into solid tissue to engulf and dissolve dead cells, bacteria, and debris. If macrophages already possess the exact infiltration skills needed to breach a solid tumor, the logical next step is to pull them out and engineer them just like we do with T cells. But they refuse to cooperate in the lab. Mature macrophages do not multiply well, resist genetic modification, and die off almost immediately. For years, this stalled out the research, until scientists at the USC Stem Cell laboratory asked a different question. What if the problem is trying to edit a biological machine that has already finished its development? To build a durable factory, they realized they had to step backward in the biological timeline and intercept the immune cell before it grew up. Looking at the stem cell hierarchy tree, researchers targeted the middle tier, Granulocyte-Monocyte Progenitors, or GMPs. Think of GMPs as teenage immune cells. Using a chemical cocktail, the team froze these GMPs in this intermediary state, allowing them to multiply endlessly, duplicating across the middle tier. This breaks previous biological assumptions. Scientists long believed that kind of continuous self-renewal was a property exclusive to the root stem cells at the very top. By locking GMPs in a state of constant division, the researchers successfully generated a scalable, living biological platform ready for genetic weaponization. With a stable supply of multiplying cells, the team inserted a CAR receptor into their DNA, equipping these teenage cells with the same cancer-detecting software used in CAR T therapy. They added a second modification called CAR-FC. This acts like a biological emergency alarm, broadcasting a signal that recruits other non-engineered immune cells into the fight. When they injected these engineered GMPs into mice, the cells migrated straight into the bone marrow, continuously produced fresh tumor-infiltrating macrophages, and successfully slowed the growth of dense solid tumors. This transformed the body's internal production line into a permanent, self-renewing source of cancer-fighting reinforcements established directly inside the skeletal system. If we compare current methods to this new approach, the contrast is stark. CAR T is a boutique process, building one complex treatment for a single patient. GMPs offer a path towards standardized mass production. Because these progenitor cells multiply so easily in the lab, they could form the foundation for true off-the-shelf medicine. Hospitals could stock a supply of pre-engineered cells, ready for immediate infusion without the weeks of waiting. These results are currently confined to animal models. Researchers still need to conduct exhaustive clinical trials to prove that these engineered cells are safe, that they don't trigger dangerous immune overreactions, and that they survive long-term in human patients. Even with years of testing ahead, discovering how to engineer the body's internal cell factories expands our arsenal far beyond T cells. It provides an entirely new strategy for fighting solid tumors. What do you think? Could mass-produced immune cells become the standard for cancer treatment? Let us know in the comments below.